All right. Here we go. All right. So one of the things that I wanted to share with you guys today is so much about what forensic science is and what it isn't, because everybody's seeing the CSI shows and seeing what, all the great things that forensic science can do. But one of the things that we have a problem with is that, sort of the reality is that we can't do everything as quickly as TV has us doing it. So, in the real world, it takes us sometimes several hours, several days to get some sort of analysis completed, and we may only have a portion of it. So this is sort of an example here for a DNA laboratory case where we would run something and we may get a DNA sequence for that, but we would only have a part of it. We don't have our suspect yet. So we may have to put that through a CODIS database. We may be waiting for them to create another crime if we don't have a suspect. So in the movies, they'll often say, they know exactly who did it. In my watching the CSI shows, I actually was very frustrated the first time I saw one and they were like, that's, the green paint is specific to the paint from the railing in Central Park. And I was just like, we don't do that. Like, it's just, we can't possibly tell you into that certainty of where something came. We certainly can help you match certain things up. And if we have a DNA profile from a suspect, we can tell you if it's included or not. But just sort of so people understand that it's not quite what CSI makes it out to be. So can I somehow lower this screen? Here we go. Sorry. Here we go. So just briefly, forensic science is really the application of science to law. So we're taking matters of law, evidence from law, from legal crimes, criminal crimes, and we're bringing them into our laboratory. Some of our cases may end up going to court civilly, but we only here in the OFS deal with criminal investigations. So no crime, no time spent at the lab. So our role, again, we're helping these law enforcement investigators, help them analyze the physical evidence that they're bringing into the laboratory. We do occasionally provide expert testimony, but it's not as often as you would think. We occasionally are brought in and we will share our results. But again, it's typically a higher end case, more like your homicide, sexual assault cases. Our drug evidence cases very rarely are going to court. Toxicology, sometimes with a driving under the influence, especially if somebody's had more than one offense, we may get called into court. And we're always training new personnel. We also do a lot of outside training of agencies to try to help them understand what our needs are. So we will do different seminars, training them on the technologies that we have, different instrumentation that we might have, and how we might be able to help them. So where are we? We have 4 main laboratory locations throughout the state. So we have our central and our DNA. Central lab is really a full service laboratory, and they're located in Hamilton, which is over near West Trenton. Cedar here is our East Regional Lab. We're located on the beach, which has all of its perks. Our North Regional Lab is currently in Little Falls, although it was recently flooded. So we are looking for a new laboratory there, which we may relocate to East Hanover, excuse me. And our South Lab is down at Hamilton, New Jersey. So we try to spread out over the state so that the agencies don't have to go very far as submitting their evidence, because we are sort of an odd shaped state. We need to have coverage so that we minimize the law enforcement when they're delivering their evidence, that they don't have to go very far. So some of the services, again, when you're watching TV, you might see scientists going out to crime scenes or doing fingerprinting, and that isn't exactly happening in the real world. Here in the Office of Forensic Sciences, we have trace evidence, and I'll go through some of the different types of evidence that are incorporated in that. A forensic serology, which is a biology unit, and then that evidence would then go on for confirmation to our DNA unit. We have the drug chemistry and the toxicology. So for the state of New Jersey, fingerprinting, crime scene evidence, evidence collection is typically done by a listed crime scene investigator. So what are the minimum requirements? So for here in New Jersey, you need a bachelor's degree with 2 years of previous lab experience. Now, that means you can't get hired in the state of New Jersey right out of college. That doesn't mean that you can't work for other states. Other states possibly have a lab tech type position that you could work in forensic science. But here in New Jersey, the salaries are commensurate with the experience that you would come in with. We are one of the highest paid forensic scientists in the country here in New Jersey. Of course, cost of living matches that, but so 2 years of previous lab experience. I'm going to go through some of the instrumentation things that might be helpful for you if you are interested in a job in forensic science, some instrumentation that you would be looking for in your job experience, lab experience, if you were pursuing a career in forensic science. In addition to a bachelor's degree, you could have a master's degree and 1 year of experience. We do require 30 credits in some sort of chemistry. So even though if you were coming in here with a biology degree, I have a biology degree, I did have the 30 credits in chemistry because a lot of things, a lot of our disciplines do require a heavy chemistry. You will get that mostly probably in your biological sciences majors, you'll have a lot of that. Now, if you're specifically looking to go into a DNA type testing, we require, it's actually a federal mandate that you have to have a course in biochemistry, genetics, statistics, and molecular biology. So if that's where you want to head, your biology degree would be great, but just know you'll need to make sure that your electives cover those courses if you want to be considered to be hired into the DNA unit. Okay. So one of the very frustrating things about having a job in forensic science in the state of New Jersey is we have what we call the Civil Service Commission. Civil Service Commission is actually just made sure that we follow certain rules and regulations as far as hiring people. But one of the things is you have to do an open competitive civil service exam. So we will, when we're interested in hiring a forensic scientist 1, which is our entry level position, we will request an opening to be filled. Civil service will post a test. So that test will have to be taken by all the applicants that are interested, and you have to score toward that top end of the test, depending on how many people we're hiring. So if we're hiring 50 people and you're in the top 50, most likely we can get to you. If we're hiring 10 people or hiring 3 people and you're in the top 10, we may not be able to get to you. So it's very important that when you take the civil service exam, that you score very well on that exam to ensure that we would be able to bring you in for an interview and be able to hire you. I have included the link here for the job announcements where jobs will be posted. I can tell you that we are not currently looking for any FS1s, forensic scientist 1s at this time, but I encourage you to go, look back and take a look here. This is where the jobs would be posted. And there's also an online, user guide that would be very helpful as far as filling out that application. And it's all online now where you could apply for a job. So again, butlers, I don't know what it is, but everybody always seems to think the butler did it. Again, I blame it all on the CSI. And how many people actually have a butler these days? But we wish it was this easy, that we could just tell the law enforcement officer, who did it. With the advances of DNA, we are certainly getting closer to being able to help them solve the crimes, but not certainly in all of disciplines, we're not able to do so. So salaries, again, we are extremely fortunate here in the state of New Jersey to be paid extremely well. So Forensic Scientist 1 here would be starting out, again, you'd have 2 years of previous lab experience and your bachelor's degree, you'd be starting out at roughly $70,000. Now, a master's degree with a year of experience would put you in a higher range. You would and max out as a Forensic Scientist 1 in the $90,000 range. A forensic scientist 2 would be a promotion. That promotion, again, would be a civil service exam that you would be required to take to be eligible to be a forensic scientist 2. You'd go through an interview process. And then we have our supervisors who run our discipline units, are forensic scientists 3. Again, depending on where we have salary ranges. The state is a union shop. So it will depend on where you are in your current salary. When you get promoted, they require that you go to at least a step advancement. We do get steps every year through a 10-step process. A laboratory director would be my position. I've been with the state for almost 25 years. March will be my 25th year. So it's not something that you typically get hired into, but it's a long range goal. And it is required to have a master's degree. A deputy director is actually a position that we're trying to create, something that we don't currently have, but we have a pending proposal with the Civil Service Commission that we're hoping that we'll have an assistant for our director. So our OFS director is in charge of all of the laboratories. So all 4 regional laboratories, as well as the DNA laboratory. In addition, we have an anthropology laboratory, and we also do some breath testing that he oversees. So just a little joke here, but we do glass analysis, and we can tell directionality. So if a brick is thrown through a window or a bullet goes through a window, we can actually just help the investigator decide which direction was it thrown through. So if you try to throw a brick from the inside and say, hey, somebody threw this brick into my house, we can actually see that. We can also see a lot with if there were bullets in a window, we can tell you which bullet went through the window first. So some of the key skills that I think would be very helpful if you chose to have a job in forensic science is we're always talking about ethical and moral character. You're dealing with evidence, you're dealing with other people's lives, and you have to follow the rules. So if you're not really a rule follower, probably not the best job for you. We have to have, the highest standards and moral character in order to be successful and carry through with the quality assurance program that we have here. Logical and independent is always helpful. I think as scientists in biological or chemistry, you have that logical mind. You tend to have a more scientific thought process. Independent is just, we work very independently. Once you are certified in a discipline, you just need to be able to kind of take those cases and work them. It's not that we don't have a very friendly environment as far as asking questions and solving problems together, but for the most part, you're going to be working a batch of cases on your own. Attention to detail is so very important. We need you to be able to pick up on things if something doesn't look right, troubleshooting, instrumentation, or taking a look at a case, something doesn't seem right, and bringing that to attention to a subordinate, or excuse me, a supervisor. Good written and oral communication. I don't know a job nowadays that you don't need good written and oral communication. We do need good written communication here. We do have to issue a report for all of our findings, and as well as oral communication, certainly with testifying in court or presenting lectures such as this. Concentration and patience. Some of the work is tedious. State government can be very tedious. I find that nothing happens as fast as I would like it to. But it doesn't mean that it doesn't happen when it needs to. It can just be sometimes frustrating. So we do ask, people to have the patience to see the process through and go through the even like the accreditation requirements that we require, attention to detail. Back to that again. Some find that the work can be stressful, and depending on your discipline, the biological sciences can be a little emotional. Finding out details of a child being murdered or sexually assaulted, personally, is a bit rough. So you do have to be able to, have some emotional intelligence and be able to work through you're having your work life and your family life. But it does take some time to get through that. And, great public speaking skills, again, with that oral communication. But we do need you to be able to not be afraid to speak in front of people. Sometimes we'll have individuals share case histories and perhaps share a difficult case that they had. So definitely speaking up in front of your small unit is not without question. So we have this wonderful theory in forensic science called the Locard exchange principle, where every crime scene we believe something was left behind. Now, criminals obviously are getting better at wearing gloves, perhaps wearing booties on their shoes. But even with that, things are still left behind. Hairs are left behind, fibers are left behind, DNA is left behind. You can touch a surface now and there'll be epithelial cells left behind. So it is hard to commit the perfect crime. So as I mentioned, discipline, trace evidence. What we do in trace evidence, we have many certifications. So if you're interested in trace evidence, it does require a strong background in chemistry. We're looking at fibers. Some of the types of evidence with fibers might be clothing fibers, carpet fibers, something if you were wrapped up in a blanket and placed in a trunk of a car, we might be able to find that blanket and then link those fibers to fibers that we collected from the trunk of the car. Fire debris, we work closely with the arson investigators in determining what type of accelerant was involved in an arson. Sometimes we can find a suspect had a can of kerosene in his car. We are then able to collect charred debris from a scene of a crime, and we can show that the fire was set by kerosene. Gunshot residue, we can help the ballistics unit do distance determinations to see if a gunshot residue is present on shooter's clothing or victim's clothing. And when it's present on like the victim's clothing, we can help them, depending on the spatter pattern of the gunshot residue, we can help them determine how far away the shooter was. And sometimes that can be relevant if somebody says they were in a struggle, and we can actually say, well, no, actually you were 3 feet away. Paints. We do a lot of paint work with car accidents. We can sometimes tell an investigation if there's like a hit and run. We can take a look at that paint, and we have what we call a P2Q database. It was developed by the Canadians, but we can actually tell them the make and model of a vehicle based on the layering structure of paint. So that can be very helpful if they're trying to get word out to an agency or to the chop shops to say, hey, look out for this car being repaired. Low explosives, we typically deal with like pipe bombs, things like that, things that might have been left behind. Sometimes nuisance crimes, a pipe bomb left in a mailbox, something like that. We're not dealing with larger explosives. That would be handled by the state police bomb squad. We do also look at hair identification, looking to see if hairs were of human nature, if they're animal in nature. We do need, sometimes we'll look at hairs for DNA, but they would need some sort of molecular, what we call a tag, but to be analyzed for nuclear DNA. We used to do hair comparisons. We've sort of gotten away from it now with really the advances in DNA being so much more conclusive. I mentioned glass. We do broken glass. We also do refractive indexing with glass, so we can tell if glass particles, perhaps somebody broke a car and then we found those glass fragments on their clothing, we could match them up. Tool marks, breaking and entering. We find that any tool would leave stria on the scene, and we can then match it up to the tool. Impressions, again, tire and footwear, leaving behind footprints or tire prints, we can match them up to the actual tire or footwear if we were able to find that. And then physical fits are always fun. You can actually take something and just like a puzzle piece, put them in, fit them in together. So just a little funny on our low explosives. So again, I wanted to make sure I touched base on with the instrumentation that we are constantly dealing with here. So microscopy in trace evidence is a big part, sifting through pieces of evidence to see what's even present. We have a microspectrophotometer as far as looking for colors. FTIR, often used comparing paints, different layer structures to see what the components are. A pyrolysis GC will evaporate the components, again, it may possibly a paint chip, and give us a GC separation. GC mass spec we use often for arson cases, looking at the accelerants. Refractive index, again, for glass. PDQ database for paints. Scanning electron microscopy is huge. So if you're interested in this and you could get experience with that, we look at gunshot residue particles, but other general unknowns using the scanning electron microscopy. X-ray diffraction, we're not using that as much right now, but certainly always good to have experience with that. And we're constantly doing chemical tests for gunshot residue. We have a lot of presumptive tests that we'll do that gives us indication of whether or not presence of nitrates. Test for metals, sometimes we're just looking to see if, depending on what direction we might be going, and also testing for oxidizing salts. So again, just give you an idea of what you might be instrumentation you might be testing if you were in trace evidence. Footprints, of course. So trace evidence, not only do we look at footprints, but tire impressions. We can often tell the investigator the size of the shoe, as well as sometimes the make and model. So it's, Converse, it has a very unique pattern. We can tell sometimes if there's very unique indentation. So you and your best friend can have the same pair of shoes, but you walk on different paths. So your shoes will actually end up being very different. So we could tell if you or your friend left that footprint, even though you may have bought them on the same day. Next, discipline, forensic serology. So forensic serology is really where the evidence comes first. We process it to see if there's any type of biological material. Typically, we are looking for blood, epithelial cells, or from a sexual assault, whether the presence of seminal material is present. So do a lot of color testing. In forensic serology, we can do a Kastle-Meyer test for blood. We do an acid phosphatase for seminal material. We look for the presence of spermatozoa, and we also do an ABA card. Luminol test is seen a lot on TV shows. We really rarely use it. So if we can't see blood, we will employ the luminol test. However, the luminol test, when we spray that all over the blood that you can't see, it actually could dilute the sample and make it harder for us to find DNA. So we'd actually prefer to take filter paper or a swab and swab the area first and try the Kastle-Meyer test. But the luminol definitely makes for a flashy TV show. A little funny for you. All right, so as I said, from serology, our evidence would then get transferred to DNA. DNA, I'm hoping genetics class, you guys are starting to talk about this, but the deoxyribonucleic acid, it is our master molecule of life, and it's the human, key to identity. So here we go. There was, when I put this presentation together, there was snow on the ground, so I apologize. But again, we can give some genealogy. We're not really doing that here as far as forensic criminal investigations, but it is on the horizon to do more genealogy work cases and telling what people's genetic makeup is. So sources primary that we're seeing here at the Office of Forensic Sciences is blood left behind, blood left from a victim, blood left from a suspect, seminal material, certainly in a sexual assault case. Epithelial cells are extremely on the rise, a lot in our property crimes. If you steal a car, typically you're going to leave some sort of DNA in that car. But again, cigarettes, envelopes, soda cups, cans, that is true in the TV shows where, they ask you to take from a cup. But there are some legalities to that. So if you have a lawyer friend, they will tell you that, they have to have a search warrant. Often you can't just take somebody's DNA without their request. So it's standing up in court might be a little difficult. You actually have to get a search warrant typically to take a buccal swab to get their known DNA. You could use that cup sometimes to say, hey, there's a reason to believe that we should get a search warrant, but just taking that and convicting somebody off of the cup that they drank out of during their interrogation is not realistic. We do tissues. Unfortunately, we do paternity testing. So if there's been a sexual assault, we will take a look at fetal tissue to see if we can then determine that somebody did, in fact, sexually assault the person. Bones and teeth, again, we work with anthropology. They submit stuff to our DNA unit from digs that they have on site, or if an investigator finds human remains. And hair, again, if there's an actual root on the hair, forcibly removed hair, we can sometimes find forcibly removed hair at a scene and link that to a DNA profile of the victim. While it may not help us find our victim, we can then say that the victim was in that location. So DNA analysis, well, it can be broken down to this 4 simple step process. It is much more complicated than that. I highly recommend you really like statistics, but we start out with an extraction procedure where we're going to break up the DNA molecules, and we're going to quantify. So it's just telling us, do we have enough DNA here to get the results that we're looking for? And if and it's sort of like combined with our amplification process. So amplification is that making multiple copies so that we can increase so that we can visibly see it, and then detection. So we're currently employing STR analysis, or short tandem repeats, for our nuclear DNA testing. So we have the repeating sequence of the base pairs. We have a locus, again, being that selective region along the chromosome. The alleles are we're looking at in our after our detection to see the different versions of the locus and see if they match up from one, from either like a suspect to a victim or an unknown to a suspect. We currently are looking at 20 different core loci. It's ever increasing. We were at 13 not too long ago, and we've already increased it to 20. So that's what the FBI is looking for, is a full identity would be, looking at those 20 different regions. But you don't need 20 different regions to have identity. For us, it's a statistical evaluation of the match. And typically the unknown exceeds that 1 in 7 trillion threshold. So DNA testing, again, being a group of biology students in a genetics class, if you can get your job experience using this instrumentation, it will help get you placed into our DNA unit. When scientists come here, we don't necessarily always place you in the discipline that you would like. So it depends sometimes on need, but we will often take a look at where your job experience has been. And so if you have a lot of DNA experience, we're more likely to put you in a DNA laboratory than to put you in a drug laboratory. And the way for you to demonstrate that is to have your job experience when you're coming in. So we have, and again, this is more specific to what we actually use here, but if you have an experience with an extraction robot, that would be very helpful, working with DNA, doing it some sort of PCR quant system, the quant amplifying, again, work with robots, doing some sort of PCR testing, using a genetic analyzer, again, doesn't matter which genetic analyzer you're using, but having experience with that. Having experience with software. We're currently using Gene Mapper software, but I can tell you, we just hired a whole bunch of people with StarMix interpretation experience because we knew we were moving that in that direction, so that we knew that they would be a great fit for our unit. So again, you having this experience will be very helpful for you in getting a job. CODIS, we have the FBI database. We use it often. We put unknown samples in there. It'll be searched against the database of previous repeat offenders. It is linked for all 50 states. Again, it does require that the 20 core STR markers in order for us to upload into the CODIS database. I don't have a slide on this, but another thing that is extremely hot and new is rapid DNA. It is something that we're rolling out in New Jersey. And while those samples are not necessarily eligible for CODIS, we have these what we call smaller ponds that the states can have individual samples looking at, where you could possibly see, like if you had a string of robberies, a person of common offender, we might be able to help get some of that information from that small rapid DNA database. Ha ha ha. All right, drug chemistry, we analyze drugs of abuse, so controlled dangerous substances, and we provide a weight and a drug. Some of the common drugs that we're looking at here in New Jersey are cocaine, heroin, marijuana, and MDA. Now I say heroin, but right now all of our heroin glassines actually contain fentanyl. Fentanyl is an extremely deadly drug. And there is some heroin in the glassines that we're seeing, but most of them contain a larger portion of fentanyl. Fentanyl is actually cheaper. There's some scheduling mistakes in New Jersey that we need to fix. Part of it is because fentanyl has a medical use. It's a Schedule 2. Heroin is a Schedule 1. So the distributors have found, well, we'll send out the fentanyl instead and get a lesser charge if they're caught with it. Drug testing, typically it's starting out with a color test. It gives us an indication of what drug we might be looking at. And then we do our confirmatory testing on GC mass spec or FTIR. We do sometimes use a UV spectrophotometer as a presumptive test. I am a Coca-Cola fan, so you can see my outrage that this person was working for Pepsi. I'm actually happy he was tested positive for Coke in the Coca-Cola world, obviously not in the cocaine world. And our last discipline is toxicology. So we're taking a look at blood and urine, and we're looking for, again, those controlled dangerous substances, but we're also looking for other non-controlled substances because you can't drive under anything. So you can't be impaired while you're driving. That is still against the law. And we also do blood alcohol content analysis to determine if you are driving above the legal limit, which is 0.08 here in New Jersey, if you're over 21. So in toxicology, again, we're doing chemical extraction, chromatography for blood alcohols, we're doing the headspace, GC-FID, we then confirm on the ELISA or GC mass spec if there's no blood alcohol present. Others, we might be looking, if there's no blood alcohol, we'll be looking at either blood or urine. We would screen those. Currently, we're using EMIT, which is an immunoassay. We're moving over to using exclusively ELISA. We've been using urine testing on EMIT, and bloods were tested on ELISA. And we have just gone through an extensive validation procedure to move everything to ELISA. Confirmatory testing, again, for blood and urine drugs is GC mass spec and LC mass spec. We are working on developing some methods here at East Lab to contribute to drug facilitated crimes. So that would be toxicology evidence, looking at blood and urine from cases that the person was incapacitated with a drug and then unfortunately sexually assaulted. So we do a little bit more extensive testing for those. That work is currently being handled in our central laboratory. We do quantitation of ethanol and GHB. GHB is a naturally occurring endogenous substance, so we do have to quantitate that, and that's done by Headspace GCFID. Love to see some ambition and tenacity. I don't know about greed, but obviously looking for really good employees. We are not drug tested here in the state of New Jersey, but it is common practice for forensic scientists to be drug tested prior to being hired. So as I said, everything goes a lot faster on the TV shows and in the movie, and everything's a lot more perfect. Our instrumentation breaks down. We have things that go wrong. And we're an accredited laboratory, so I can tell you our quality is good, but we just can't possibly do it the same way that they do it on TV. And to give them credit, I truly believe they try to get it right, but they have to make a living. They have to sensationalize it a little bit. They have to make it so that somebody's going to want to watch the show, and they can't, show you that it takes 3 months to do a case. because nobody wants to watch the same show for 3 months to hear what happened. So I do understand that. But the problem that we've had here in forensic science is juries now believe everything should contain DNA evidence, or that everything should be able to be solved in a nice tiny little bow wrapped around it, the way that the CSI shows have demonstrated. And so we actually will sometimes have to testify just to explain to a jury, what it is and what it isn't, so that they can fully understand what our capabilities are, and that the TV shows don't exactly represent the truth. So I did want to spend some time talking about my journey, here through, and I think we've got on time. But so when I was growing up, I wanted to be a detective. And my uncle was a police officer, and he laughed at me. He said, "You don't get to be a detective." And I'm like, "What?" He's like, "You have to be a police officer." And I was like, "I'm not handing out speeding tickets." And I wanted, to be a crime scene investigator. I wanted to solve crimes. I'm old enough that I was watching Quincy growing up. And, I wanted to be Quincy going to crime scenes. And, I didn't know that medical examiners don't really typically go to the crime scene. So I was like, I want to be the detective that, solves the case. I was really good in math and science, and my mom did not really want me to be a police officer. So I pursued biology in college, and had thoughts even of possibly becoming a medical examiner. And my uncle, again, who was a police officer, introduced me to someone who was doing bloodstain pattern work. And I was like, oh, I didn't even know this. And this was at a time before O.J. Simpson. This was a time when people didn't even know what forensic science was. It was not very well known. There were very few programs out there. There were even fewer master's programs. And so I was like, what is this about? It definitely was my way of being like, I want to, be involved in that law enforcement side, and I can use my skills, in biology. So for me, it really just seemed like this perfect marriage of being able to combine science and law. So I researched forensic science programs, and again, there really weren't that many available to me. So there was New Haven up in Connecticut, John Jay in New York, and I think there was another school down in Alabama that were really the forerunners in a Master's of Science program in forensic science. So I attended John Jay. I was not impressed, and I'm not trying to bash John Jay. They've done a lot of work since then, but I come from Rochester Institute of Technology. It's a private institute, and they had a lot more money than John Jay. So I was a bit, shocked and disillusioned to go and get my master's program, my master's degree there. But I learned a lot about the foundations of forensic science for them. And some people will come to me and say, well, why not get a degree in forensic science? And I don't recommend it. And I will tell you why. I believe you're getting a degree in a biological science or in a natural science, chemistry, biology is so much better, and you will learn so much more core science than you will in forensic science. So forensic science, they'll teach you in a master's program, we'll teach you on the job, but we need to make sure that you understand the fundamentals of science, of chemistry, of biology, in order for you to be a good forensic scientist. So I feel that the forensic science undergrad programs sometimes can be a little weak. It's a generalization. I'm not saying all are. I know some very good ones, but in general, I prefer you to have a biology or a chemistry degree or something close to, you could major in molecular biology, whatever, that type thing. But I think that provides you with a really strong background. So I went
Laura,
to-
excuse me, I don't want to interrupt yet. We have about 5 more minutes until questions.
Okay.
Right. Thank
you.
You
Got it.
This is great. Continue on this path.
Yep. So I worked in a pharmaceutical while I was waiting for a job. As I said, we're a civil service organization. So unfortunately, we don't hire all the time. So I went and worked for a pharmaceutical, and I got a lot of really amazing job experience. And that's how I got my job with the state. I had worked on GC mass spec, I worked on LC mass spec. And so, they saw that on my resume and they were like, ooh, this is great experience. So I was hired into an equine, that's horse testing. We no longer even do that with the state police. It was not where I wanted to be, but it was my first chance. I was finally working in a forensic environment. Really quickly transferred to one of the North Lab up in Little Falls, and was able to start in our drug chemistry unit. And I'm like, woohoo, again, not where I wanted to be, but I was willing to put the time in. I learned a lot about drug chemistry. Because of my previous lab experience, I was promoted really quickly to a forensic scientist too. And I was able to actually train everybody else. It was instrumentation that I had used at my previous job. We were just getting new instrumentation here. I was able to train people on that instrumentation. Finally get transferred to trace evidence at the central lab. And I'm like so excited that I'm finally going to work, in the job that I considered, a dream job when I was going to grad school, only to be pulled back into drug chemistry because that's where the backlog was. So I went back to drug chemistry, only to find out that toxicology was having some issues working on the drug facilitated sexual assault. And they needed someone who knew how to run an LCMS. And guess who had that experience from a previous job? So I went in and I helped develop methods to identify these low levels of benzodiazepines for toxicology drug facilitated sexual assault cases. And again, I still wanted to get back to trace evidence. So my bosses thought it would be, we'll transfer you to serology. At the time, serology and trace were in a group called criminalistics. So I think they kind of thought, we'll throw her a bone, we'll put her in serology. And again, loved it, really learned a lot. I was a little closer to evidence that I found fascinating, and I worked in serology, and I finally was able to get into trace evidence. And I was training, I worked a lot of hair cases. We did hair comparisons back then, worked with fibers, worked with paint, and I really did enjoy it. I was also assigned to be our LIMS administrator, which is our laboratory information management system. And I learned a ton of information from that. I didn't volunteer for it, but I was, they wanted me to do this. So I was like, okay. But I learned a lot from it. And later on, it became very important that I learned that information. And then suddenly a promotion became available, and it was in serology. I never wanted to work in serology, but thank goodness they had asked me to get trained in serology, because now when this opportunity became available, I was able to go be a serology supervisor. So I was a serology supervisor. I then get voluntold. We had some issues going on in our evidence receiving department that needed, someone to be sensible. And apparently I fit the bill. I didn't really want to go to evidence receiving, but I was voluntold, we need you to go up there. So I did. And I learned so much in our evidence receiving department. I brought that with the LIMS administrator responsibilities that I had. And I learned to have a great respect for the officers. I learned what they needed, what they wanted. And so when I was done kind of shaping up the evidence receiving unit, I was able to come back to serology, and I had all this information that I knew that the police officers needed from us. And so I was able to implement some changes and some review processes that really made a huge difference in the organization. And with DNA, I was able to become promoted to an assistant lab director. I then came out here as an assistant lab director, as a lateral, knowing that the lab director here was going to be retiring, and I've become the DNA lab director. I certainly aspire to be the deputy director when that position becomes available, and hopefully one day I could even be a director. So I wanted, sort of to share with you just this crazy path. It was never a straight path. It was never, I wanted to do this and then I got to do it. And I really want to encourage you guys to be willing to zig and zag and to be able to take up those new opportunities and to be, pushed outside your comfort zone and to take on things that other people think that you can handle, because it's that zigzag, and you never go up a mountain straight. You have to kind of do that back and forth, up the mountain. And I for me personally, it's really paid off. So that's it. Did I do okay? Time-wise?
You did wonderfully. Wonderful. Thank you so much. I thank you, everybody. Join me in thanking. I know if we were in person, we'd be clapping, waving hands, whatever we do, maybe putting, emojis on our screens.
Yes.
But thank you so much for your real good, deep look into forensic science and your personal journey. There are so many questions that we have from our students. And I'm just going to summarize a few of them. I know we won't have enough time, so I've been trying to look on this other screen that has all the questions coming in and sort of hopefully I'll get the gist of the combined a few. Right.
Thank you.
So we have ones that are coming in a few different areas. And so there's a few questions that are related more to the process of a crime scene investigation, about how the lab that you work in collaborates with detectives, and how do people who collects the data, who consults, is there a coroner involved, how does the whole larger team work, and what is your office's or lab specific role in that bigger crime investigation setting?
Okay, so for an example, if there is a homicide, there'll be a first detective on the scene. He will call for a crime scene unit. Again, here in New Jersey, those are enlisted either for state police, would be a trooper, or a law enforcement officer would then go. But they're people who are, it's actually by law, you have to be required to have received training in crime scene investigation to respond to that scene, to collect the evidence. So they would go through that crime scene and try to collect what they think, and they have to be all encompassing because you only get that one chance before they release that scene. So they will collect evidence. They'll bring it back to their agency and start to go through it and start with their questioning of witnesses. And then they will bring that evidence, not all of it, because they might collect 200 pieces of evidence. They're going to try to break that down to possibly what they think might be the 10 most relevant pieces of evidence for us to take a look at the lab. So they'll submit, basically it's a pre-submission form, and they'll kind of tell us what they're thinking. We review that, the evidence gets submitted to the laboratory, and then depending on the type of evidence, it gets brought into that discipline, and it would get assigned to an individual analyst. Now I can tell you the only area that we, well, there's a couple areas that we do assist at crime scenes. We don't go to a lot of crime scenes, but again, with that luminol, as I mentioned earlier, if an investigator can't see blood, like they're like, I think somebody was killed here, that it looks like maybe there was a cleanup, but we can't see it, we'll go and we'll try to do some swabbings. And if we can't see it, we will spray for luminol. That is something that we will do from the lab. Some of the agencies are starting to do a blue star reagent, which is similar to luminol. So they're doing that on their own. But some agencies will still have a scientist come out and help them find blood. We also will respond to scenes as far as shooting reconstructions to try to help if there was like a car involved. If we go out and they're doing some, they use all kinds of rods to try to figure out what direction and what bullet sequencing there was. We'll actually try to help them sometimes determine if there's gunshot residue present. Does that help answer the question?
Yes, that's great. The one other question that was sort of wrapped in there was the role of the coroner's office.
Oh, yes. Okay.
How does that
so
work?
Here in New Jersey, we don't have a coroner system. We have a medical examiner system. And it's different in that the coroners are elected official, almost like a sheriff. So states do have that system. Here in New Jersey, we have a medical examiner's office. Medical examiners here in New Jersey do not respond to the scene. Coroners in other states will actually respond to the scene and declare somebody dead. But medical examiners here in New Jersey, to the best of my knowledge, are not going to the scene to declare somebody dead. They will respond. So the body will be brought to the medical examiner's office, and they will do their examinations, and they will take blood and their own toxicology analysis. So if somebody is dead here in New Jersey, we do not get the toxicology evidence in the lab. We only do live evidence, antemortem evidence here. Postmortem toxicology gets done at the medical examiner's office.
Okay, great. Thank you. I think that answered a whole slew of questions that we had. And this one also kind of combines a few different questions, so bear with me. Depending on how the student asks the question, it's essentially, what was your most difficult, most tedious, or most interesting case, right? Obviously, without giving away anything that you can't talk about. And kind of rolled up into that same sort of question is, how long does it take to go through a typical case, like collect the evidence, process the evidence? What may make a case more difficult than another, right? So they're looking for that timeline, as well as what makes a case maybe really interesting or really difficult to handle.
So personally, one of my most difficult cases, I really had problems with children. And when I had my own children, it became even more difficult. I was working in the serology unit, and you would typically hear, sexual assaults. There would be murder cases, but one particular case I had a little post-traumatic stress from, honestly, and I wasn't even aware of it. It was sort of through training that I became aware that this even bothered me. But I had an instance where a little boy was asked to shake somebody's penis. And we got the evidence into the lab, and I just read it. I didn't go to the scene. I was very distanced from this, but just reading that. And so, that's where I say that some of this stuff can be very emotional. Just reading that really disturbed me. And I ended up having a nightmare about it with my own son, us going to a restaurant and having a waiter ask him to do the same, and woke up just, horrified. And it was very disturbing. That was the only time that that's happened to me. I've heard of other scientists having, sort of that. It's really where we're very trained to think very left brain, be very logical, very analytical. And every once in a while, your right brain sneaks in and you get a little, emotional, and you make it personal. And part of our job is that I can tell you, I can look at bloody scenes and pictures of things, and for the most part, I see the science and I'm looking for, what am I going to do with this? And what am I going to do with that? But every once in a while, that emotional response happens, and we have to deal with it. We do have a wonderful employee assistance program here that we have counselors available for us to talk to, not only work-related things, but even if there's something happening outside. So that, is wonderful, part of the state police organization. Allison, I'm so sorry. Second part of that question after me was how long? So typically it takes a good year of training to even become certified. And then even when you get cert, so you have to go through some training samples. We will create like live samples for you, but that are not actual casework. As an accredited laboratory, you can't touch real evidence until you're certified in that discipline and have been authorized to work. So we have you follow somebody through the discipline first, really just kind of have them showing you what they're doing. And then we'll have you start demonstrating it with training samples so that you can get a feel for how things work. So we have you sort of watch, then demonstrate, and then show that you're proficient. And then we give you a certification exam. So it really depends on the discipline and your previous experience on how long it will take. Everyone is different. Some people learn quicker. Some people, take a little bit longer, and it is very personalized. Your training is all about when you're ready, when you feel that you're, able to do what you need to do. Sometimes we have to push, and it give you a little boost of confidence that we believe in you. But it really is individual. We would like to say a year, but like trace evidence has all those different components within the discipline. That's a year for each one of those. So you might take you a year to get certified in DNA. If you have previous training in DNA or if you came from another lab that has, they've been doing DNA testing, the training will be a lot quicker. Where you were working at an aquarium and now you're going to come in and we're going to train you as a drug chemist, we might have to do a lot more work. So it really is individually based.
Thank you so much. Based on that, I think you've already gave good advice about training and experience that you would need. But if we have some students in our audience right now who really do want to be forensic scientists and they're biological sciences majors, which is what I think most people are, they may be biochemistry majors as well. What sort of pieces of advice do you have specifically? You've already given some great advice. Is there other specific next steps? Like, what is that first job to get that 2 years experience? What's the best places to be looking for those jobs?
So you really want to be getting that job experience, most likely at a private lab, or going to a lab that starts out with somebody who's right out of college, like the Office of the Medical Examiner in New York City, referred to as OCME. They will hire scientists right out of college with a biological sciences degree. Now, again, making sure you have the core requirements for DNA in molecular genetics, statistics, and biochemistry. So, those 4 courses are a deal breaker. And a lot of master's programs now will even require you to have those to be matriculated into their program. But if you're seriously interested in working in a DNA lab, you have to have those as electives. But then I would say, trying to find a private DNA lab, or sometimes, like I said, needing to leave Jersey to find a lab where you can go be. Sometimes they'll hire you as a lab tech or a trainee or biologist trainee. Some systems have that. Unfortunately, we've just simplified it in New Jersey, and we don't do that. And we've just made it so that you could make a living wage, if you were working here in New Jersey. But that's the key, is to find a lab that you're going to be using some of the instrumentation that you would be using here. So you're gaining that experience, genetic analyzers against StarMix. So you have that experience, and it'll make your training go a lot quicker. So you'll be able to contribute to the unit faster.
Great, thank you. One last question, and then we're done. And I think you've already touched on it a little bit, but so what aspects of forensic TV shows are most accurate, and which ones are not at all? Like, what's like, if you have one thing that you say, like, yeah, they get this right, and two, they totally get this one wrong?
So there's different types of crime shows. So some are truly just entertainment, but there's actually a whole bunch of series like the Forensics Files that they're actually doing a review of real cases, and they're getting it right. They're telling you what happened. So that type of show isn't really about, they still, might put some lights and, color and stuff like that on it, but they're talking to the real investigators, they're sharing with you the real evidence, and they're walking you through what was found. So those are very, very realistic. The CSI shows, it's really, sometimes it's a matter of they use the wrong instrumentation, or they truly oversimplify the testing so that they can find a result. So the joke is, that the butler did it. Or the example, like I said, so in CSI, they were saying that they would ran it through GC mass spec. They found out that it was green paint from Central Park. So I can tell you, paint analysis isn't typically done with GC mass spec. We would probably do electron microscopy. We would do a microspectrophotometer for color. We would do matching up of the different paint layers. We would do FTIR, looking at the individual components of that. So again, GC mass spec is typically used more for our drugs and tox. And we use GC mass spec for fire debris, not typical instrument for paint. Every viewer would not know that. So could we find out that we would see that there was green paint? Could we find out the components of that paint? Yes. With car analysis, we can find out make and model. But typically we can tell you if it's a household paint, but I can't tell you it's Benjamin Moore paint versus Sherman Williams, paint kind of things. So I think the TV shows are trying to get better. They actually are hiring forensic consultants to try to help them with, some of the DNA testing so that they can help make it more realistic. But I can tell you the same thing happens for the doctors or the lawyer shows. If you have a doctor or a surgeon who watches, Grey's Anatomy, they're like, oh my goodness, that's, that doesn't happen, or that, that person can't, a nurse can't do that, or a doctor, whatever. So I think it's just the nature of television. So I would take it as enjoy it, but know that they make everything happen a lot quicker. And there may be several steps involved in an analysis that they do much quicker. Actually, the very first question you asked me was like, how long does something take? And that is very dependent upon the case. A drug case we can get done in a day, honestly. A tox case might be a week, but that's because we batch things as far as extractions and confirmations. DNA testing, again, probably we can do it. We could do a case in a day. It wouldn't be efficient because of the way we do our batches. But it all depends on what you have in a case. Do you have a victim? Do you have a suspect? Do you have that buckle reference for us to know, what your profile looks like? Trace evidence, you have to sift through sometimes piles and piles of what we would think of like, think of a vacuum cleaner, pieces of lint that you're looking for a piece of glass or a hair or a paint fiber. So it can take a lot longer to go through. excuse me. It also depends on how much evidence is present. We try to limit the amount of samples that we test, and just for throughput. But sometimes we don't find the answer in the evidence that we originally looked at. We have to go back and look, keep looking, keep digging, keep trying to find something until either we've exhausted all the evidence or we found something that we could move forward with it.
Thank you so much. So this was wonderful. This was very informative. I'm very happy that you were able to join us. So thank you very much. And thank you to
for
our
having me.
speakers for the great questions. Remember, there's more seminars coming up, so be looking for the flyers around the building. Thank you all for coming.
Great.